a professor at Clemson University. Dr. Alexov's work has been focused on understanding the molecular effects caused by genetics, specifically DNA variants of different diseases, and of particular interest are monogenic diseases and X-linked disorders. Very appropriate for this forum. And so Dr. Alexov's talk is titled Computational Analysis of Missense Mutations in the Creatine Transporter Protein. And with that, Dr. Alexov, if you're here, you can go ahead and unmute yourself, turn on your video, and please start sharing your presentation.
Okay. Can you hear me?
Yes. Yes.
Okay, before I present whatever I have to present, I'd like to say several words about our work. First, we are very beginners in this type of disease. We started the work perhaps less than half a year. Unfortunately, many of the things that I anticipated to achieve were not achieved due to many different reasons. And if I have to speak about reasons, that will be another talk. So consider my talk as a kind of, I would say it, preliminary results. I don't consider myself specialist in creatine deficiency syndrome. And many of the things that I'm going to say today might change tomorrow after we complete this study. So our lab is computational lab. We don't do experiments. And we decided to study this protein with the same way as we study other diseases.
Dr. Alexov, I'm so sorry, but if you're sharing your screen, we're not able to see it. So could you please try that again?
Do you see it now?
Yes, perfect. Thank you.
Okay, so I'm skipping the introductional part. The way how we typically approach any disease associated with missense mutations, we try to find as many possible clinically annotated mutation and their clinical relevance. So in case of creatine transporter disease, we went to ClinVar and we collected all mutations that are missense. And we noticed that in that case, we're getting about 94 missense mutations, most of them not classified or having conflicting annotations. So in most of bioinformatical, biophysical studies, when you have a database, you like to have equal population of cases. Say, take a look at the table that I'm showing right now. In different cohorts, I show different clinical annotated variants, pathogenic, benign, and non-classified or conflicting informations. So among these 94 mutations, 64 were not classified. That if we take the data as it is, that will be very unbalanced and set. Second thing, if you work with ClinVar, perhaps you've noticed that ClinVar is a database that in many cases provide information that is not extremely believable. And there are quite many false positives. So in order to reduce the noise, we decided to restrict our study with the cases for which at least one publication was linked to this particular mutation. Let's say the first one, P54412, will be included in our dataset only if there is a paper related to this mutation. So at the end, from 94 mutation, and that was about 6 months ago, we ended with this short list where the population or number of pathogenic, benign, and unclassified is more or less balanced, similar to each other in number of cases. So as previous speaker, Quar, I hope I pronounce your name correctly. We noticed that there is no structure of human creatine transporter protein. And we did similar stuff. We also do a lot of homology modeling. We do a sidebus to identify the best template based on the sequence identity. And in our case, we selected this protein. As previous speaker indicated, there are quite many proteins which structure can be used as a template, but we decided to use this one mostly because of the sequence identity, which is pretty high. If you do a homology modeling, perhaps you know that when you get a sequence identity about 40 and 50, more or less you're quite sure that the model that you produce is quite good. That was our motivation of choosing this template. So after producing the model, and for that purpose, we use a Swiss model, we came up with this structural model, which is shown on green on the left, and then the model is superimposed. And then the model is superimposed on the template, and the template is shown with red. Again, if you're in the business of structural modeling, you'll be pleased to see a very nice alignment of helixes. Of course, some of the loops and some other elements that didn't have a template might not be so believable, but at the end, the overall fault, more or less in our hands, seems to be okay. So the thing that is very troublemaker for modern computational modeling is the fact that membrane proteins do not exist in water per se. They are embedded in a lipid bilayer or lipid membrane. So to model the conditions in the living cell as much as we can, we decided to embed this structural model into a lipid bilayer. And that was done with a bunch of software, VMD, and other things, plus manually repositioning the protein, such so to have kind of good model that based on the physical chemical properties of the protein, hydrophilic residues are outside the membrane and hydrophobic residues are inside the membrane. And this is a side view, and this is just the protein without the membrane, but with water. And I will speak about creatine and sodium ions later on in my talk. Another thing that typically we do before doing any computation of energy is to have a kind of structural view where the mutations are, and from there to make some kind of guess. Do we expect these mutation sites to be important for either stability, binding, flexibility, you name it. And here is a cohort code where the pathological mutational cohort in blue, this is one, this is another one. Benigns are cohort with green and unclassified with cyan. Perhaps you like to take a look at several cases. For example, a pathological mutation, pathogenic mutation, is very close situated in the structure with mutation classified as benign. So based on that, it is very difficult from structural perspective to say if mutation is causing disease or not. I'm going to stop here for a moment. Maybe I'm going to use this slide as well to make a link with the previous speakers and to express what is our opinion about effect of mutations. As a biophysicist, molecular biophysicist, typically we infer the disease-causing effect via some changes of thermodynamical properties of protein. Proteins function as they adopt particular fault. If the fault is if the protein doesn't fold correctly, most probably the protein is not going to function. And we assess this possibility by computing what is the effect of mutation on stability, which is inferred as a change of the folding free energy. The list of possible molecular effects can be extended to many other things, like if the protein has a partner, how the mutation affect protein binding, protein RNA binding, DNA binding. In this case, how the mutation affect the binding of creatine and the transportation of creatine across the membrane. Furthermore, because this is a membrane protein, mutation effect that causes disease could be related to the fact that mutation does not allow protein to enter the membrane to be stable there. We cannot address all these questions on a large scale, and especially in this case when we decide to study creatine deficiency syndrome. We don't have much experience with this protein, so we decided to run the obvious task, namely to predict what is the effect of mutation on stability. And in that case, we use our software called SAFEC plus third-party software. How they work is illustrated on this graph, where the folded protein is shown on the top, either the wild type or the mutant, where the mutation site is shown with a yellow star. And we like to assess what is the folding free energy of the wild type, of the mutant, and then to find what is the difference. We developed quite many methods and software to addressing this question, and one of them is SAFEC. I don't think that I have to explain the equation. The equation is based on the presumption that the energy, or in that case, the energy change, is a linear combination of different energy components. If you're interested, I'll go over that, but essentially, the folding energy can be decomposed into van der Waals component, power component, non-power, et cetera, components. And in order to assess the precision of our method, typically we first benchmark it against available experimental data. And these are the coefficients W0, W1, etc. They're undefined in the initial building of the model. When we benchmark the model against a large data set, for example, taken from a ProTerm database of several thousand experimentally determined changes of the folding free energy, we adjust this W1 and 0, 1, etc., such so to obtain the best fit. And once the best fit is obtained, we use this energy formula for assessing the effect of mutation of unknown, not experimentally measured quantities. This is fine, but here we are dealing with a protein that is a membrane protein. Neither of existing methods take into account that the protein of interest is a membrane protein, and the membrane is present more or less in the experiment, and definitely is present in human body. So to address this question, we use so-called MMPB-SA method, which stands for molecular mechanics, Poisson-Boltzmann surface area. Again, all these energies, they're very well defined in computational biophysics community. I don't intend to speak about these energy terms today, but I'll be happy to get questions. And this this diagram shows how these energies were calculated. I use a generic representation of protein, but in our case, the folded protein was surrounded also by a membrane. The folded protein was surrounded by a membrane, as it is shown over here, and the membrane was explicit membrane. The waters were present as well, the corresponding ions as well. So then we compute what is the energy of folded, energy of mutant protein, and in order to mimic the unfolded state, we use a approach that we and many other people use for simplicity. We simply cut a segment of 3 residues around the site of the mutation. And then subtracting these energy terms, we obtain what is the change of the folding free energy caused by the mutation. On the next slide, I'm going to show you the results of our modeling. These are the mutation that I show you at the beginning of my talk, and the color code is again the same, with kind of reddish color pathogenic, with the yellow benign, and greens are non-classified mutations. And with vertical columns, I show what are the results of the predictions done by different type of software. And everything that you see here are folding free energy change in kilocalorie per mole. And if you see something that is positive, that means that the prediction is that the mutation makes the protein more stable. If you see something that is negative, that will indicate that predictions is made that the mutation will make the protein less stable. And all these four methods or web servers, they do these predictions without presence of the membrane. Here are the predictions made by us using MMPBSA method that takes into account the presence of the membrane. So if you browse this table, the first observation that perhaps you're going to make is that indeed pathogenic mutation more or less are predicted to cause significant change of the folding free energy. However, at the same time, benign are predicted to cause much less. And it is mixed for unclassified mutation, which give us the opportunity to reclassify these non-classified mutations. I'm going to skip that thing for slightly later. I don't like to discuss this number very much. I can show several cases for which you simply scratch your head and have no idea what to say, like this one, or even this one, where one prediction method indicates that the mutation is stabilizing, other are predicting it's not stabilizing, etc., etc. So all these methods intend to disagree with each other, but we are very happy when we see a consistent prediction, as for example, this one, that will give a high confidence that indeed this particular mutation will make the protein less stable. No matter what method is used, the prediction is consistent. Perhaps you're going to see at this part of my presentation a very long list of residues and some other number. And I'm going to get to this thing is sometime at the end of my talk, which is approaching at this stage very rapidly. So I like to make a small summary of what we found so far. I don't know how to get out of this icon. Like that. So more or less the results of the web servers and the results of MMPBSA minimization method are more or less similar. And the thing that we saw on the previous slide is that indeed pathogenic mutations are predicted to affect the stability by a larger extent than benign mutations. And many of the unclassified mutations may be reclassified based on predicted effect on the binding free energy. So, as I apologized at the beginning of my talk, we were unable to achieve most of the goals that were planned due to different reasons. However, I like to show you something that make me quite optimistic that in near future we'll be able to finish the work. Namely, we decided to try to predict what is the pathway of the creatine across the creatine transporter. So for that purpose, we positioned our creatine, shown here with red balls, in a pocket inside of creatine transporter that we believe is kind of good guess. Again, I'm quite new in this area, and perhaps many of you have better understanding of where the binding pocket should be and how the creatine should bind. But we made this model based on the human brain type creatine kinase, where the creatine was structurally, experimentally seen in the crystallographic structure. We've noticed what are the specific interactions, and then we attempted to position the creatine inside the human creatine transporter such so the same interactions to be present. And then we use something that is called steered molecular dynamic simulation, such so you apply force in direction, if you follow the cursor, in direction from here to there, and ask the molecular dynamic simulation to push the creatine direction downstream. And I'm going to show a movie that is really very preliminary results that will indicate how maybe the creatine moves across the creatine transport. So at the beginning, the creatine stays, trying to find the best orientation, makes a small step downhill, stays again in another pocket, sample a lot of conformation. If you pay a close look of what is happening, the protein more or less is stable. There is some small structural arrangement. And I gotta tell that everything that I'm showing right now was done in the presence of the membrane and explicit waters. For simplicity, they are not shown in this movie. As we see, the creatine progresses its release to outside of the membrane. And as you perhaps notice, this trajectory is kind of stepwise trajectory. There are pockets where the creatine stays a water bit, then moves in a kind of giant step, gets to another pocket, etc., etc. We were unable in the time frame, I apologize again, to run enough long-term molecular dynamic simulation to let the creatine get out. And currently we are doing so. The result of such an analysis, and it is really very premature analysis, can be used to identify which residues are in contact with creatine as creatine moves across the creatine transporter. And that was the thinking that I was attempting to show you over here. These are all residues that are found to be interacting with the creatine as the creatine moves across the creatine transporter. And here with percentage, we show with what is the fraction of the time this particular residue interact with the creatine. The larger this number is, more interaction this residue bears with the creatine as creatine moves across the creatine transporter. The circles indicate that some of these residues are also shown in ClinVar. With red circles are residues that are listed in ClinVar as a known pathologic significance, and only one of them, the cysteine, is listed as a pathogenic. So here is this cysteine, which if it is mutated to tryptophan, is listed as pathogenic, and there is a paper that confirm such a notion. But if you take a look at the numbers, some of the predictors show that this mutation should destabilize. Some predictors predict it should stabilize the protein. The numbers are not so big. So based on that, I'll be having troubles to classify this residue or this position as a pathogenic. But if I take a look at the fact that this cysteine is important for the transportation of creatine, that will give me a clear view or clear indication that perhaps this is pathogenic residue. And I like to acknowledge people who did the lab. Angela, she just became a mother. And this is one of the reasons that we were not able to finish the work. Mahesh Koraila is a graduate student in the lab doing molecular dynamic simulation of creatine. And Mihiri, she does structural modeling. And thank you for your attention.
Thank you so much, Dr. Alexov. That was a great presentation. Dr. Kolis and Dr. Alexov, okay, great. You have your videos and your audio on. I'd like to welcome Dr. Stiles and Dr. Schlebach to moderate this session. A quick note for all of our audience members. This session is going to be moderated by Dr. Schlebach and Dr. Stiles, both of whom are on the research side of things. I do want to quickly address what this type of, the implications of the kind of work that Dr. Alexov and Dr. Kolis have been focusing on. So these types of studies help us understand how mutations impact the function of the creatine transporter. The structural studies also help us understand what kind of drugs could be used to rescue the function of transporters. And so with that, I'll let the moderators take over. You have questions in the Q&A box, so feel free to take it away.
Sorry. Should I start?
Yeah, go ahead. I think you had a couple of the questions yourself.
Yeah, I'll start with mine, and I'll read up as we pivot to you, if you don't mind. So Claire, I have a question for you first concerning your model where you're docking in the different creatine analogs. If I saw correctly, it looked like the carboxylate group had a CH hydrogen bond with the glycine backbone. Is that what your model suggests?
Yes, that's what it suggests. Yes, exactly.
So, I mean, I guess what I'm wondering, I mean, I know if I remember correctly, the KM for this, it binds creatine fairly weakly, right? High micromolar maybe. And I guess, you know, if you're thinking about developing pharmacochaperones, you want something that's going to bind very tightly because it's going to push the stability. So I'm wondering, you know, based on your models, if you could, if putting different sort of groups in place of that carboxylate might help improve packing. Have you looked at that at all in terms of removing that group or replacing it?
Not yet. So I would like to say yes, that the first step of this work was just to sort of validate the model by rationalizing ligands that are already known, and then the next step would go into a little bit of more unknown and trying to find new drugs or trying to replace some groups as you suggest. But personally, I think I would try first to modify the other side of the ligand, the guanidine side, because based on the other members of the SLC6 family, it seems that the interactions in this area of the binding site of the carboxylate with the glycine that you were mentioning, and also it interacts with the iron, it seems that it's a region that is essential for binding. And so if we modify it, maybe we will lose complete binding, and so I would try to go the other way and try to, because there is room for adding some moieties there, and so maybe add moieties that would, yeah, exactly, strengthen the interaction as you suggested, but on the other side. Yeah.
There's more room to build, I guess, on the.
Yes.
And then I had a question from Emil. You sort of answered my question. I was curious about how much of your stability predictors were optimized for membrane proteins, but I suppose your approach doesn't really care as long as you've got the solvent in the simulation or in the model. I was curious though, so you have a list of mutants, you've made your predictions, and it's clear that your stability predictions are better than some of the other canned algorithms. But I guess I was curious if you put this in like a receiver operating characteristic, what sorts of discrimination you're getting in terms of false positive, true positive when you're trying to classify these variants.
And in this case, we didn't do a receiver operator characteristic curve simply because there's few mutations. So any statistical analysis on 20 cases is statistically insignificant. So I cannot address your question in this way.
Yeah. In that regard, there actually are, I think, some 75 mutations in the LOVD LOVD database. So I'd really be interested in seeing sort of how it performs with a larger set, because this is a problem, as you know, that is a larger problem across many diseases, right? And this could be an interesting case. One more question. The steered MD, is my understanding that the transporter alternates between sort of outward and inward conformations. When you push the creatine molecule through, do you see any significant changes in the conformation of the channel, or does it sort of stay more or less constant?
So everything that I said today is preliminary, really very preliminary. On the video that I show you, perhaps there is no obvious conformation change of a large scale.
Okay.
Of course, helices move a little bit, something is happening, but nothing dramatic. But any molecular dynamic simulation must be repeated several times,
yeah, sure.
because everything depends on initial velocity, etc. I'm kind of hesitant to draw great conclusions from just a single molecular dynamic run.
Yeah, yeah, and in this regard, we actually have models in multiple conformations. If you're interested, we'd be happy to share. Because there's something like 25 templates between all the LUT structures, the DAT structure, and the CERT structure. So if you're interested in playing with this more, just let me know, and we'd be happy to give whatever we can to help.
Actually, I took a big risk presenting this work because it's just a beginning work. And the reason for taking this risk is just to see you guys and get some insight of your work and become part of this community. So I'll be reaching to you definitely.
Yeah, absolutely. I think the interaction is very valuable, and you know, I like presenting preliminary data in a forum like this. It's good. Anyway, sorry for the audience members who are not interested in modeling. These are sort of technical questions, but I think it's very good work from both of you. So I'll pivot to the other moderator.
A question for Claire. Can you design inhibitors of the creatine transporters based on the homology model observations that you're finding?
Yes, actually, that's one of the goals, is to use the information we gathered from this first study of rationalizing the binding of known ligands to find new ones. So for example, as I just discussed, we know, and also based in the context of the entire SLC6 family, we know that the area in the carboxylate is essential for binding. So now if I screen a library of drugs or new compounds, I will focus on the one that interact in this area of the model and try to find new compounds that would maybe have potential for increased interactions. So yes, that's one of the goals exactly.
And question for Emil. Have you tried the same analysis with deletions instead of the missense point mutations?
Deletions are not easy in terms that if they occur in a loop, we have refinement methods and that could be done quite well. Deletion in a helix or a beta strand is very difficult to model. You don't know. I'm a computational guy, but I have quite a reservation of modeling gaps in a secondary structure elements like helixes and beta strands. I won't believe my results much.
Okay.
I think there was another question from Amaya Kester about, for both speakers, asks whether there's more than one binding pocket for creatine, the creatine transporter. I'm curious if your model suggests that there's much wiggle room for the molecule in the pockets, or whether it's pretty tight.
Yeah, so maybe I can start. In my opinion, yes, because there are a lot of studies going on the prokaryotes, the leucine transporter and the serotonin transporter, that there are some other sites. One seems to be in the upper part of the transporter, that is, it has been shown for the serotonin transporter, but there are also other potential sites such as lipids, protein-lipid interaction sites. And I think that all of these sites needs to be explored and investigated to try to find new ways to target the transporter also in a more specific way, because it's likely that these sites are more specific than the main orthosteric sites. So that's one of the paths I would like to go to also, yes.
We're also planning to do exploration of different possible binding sites and pathways by positioning the creatine on the top of the creatine transporter protein in presence of membrane and randomly sampling the space on the top of the protein, applying against tiered molecular dynamic simulation to see if different positions will result in different pathways, or they'll be focusing on the same pathway all the time. We have several papers on different proteins, for example, spermine synthase, where we position the ligand far away from the protein, even on opposite side of the bionic pocket, and steered molecular dynamic simulation deliver the ligand to the binding pocket despite the initial position of the ligand.
And another question for both of you. Have you tried to model any potential gain of function mutation in the creatine transporter receptor?
Personally, not yet, no, but it's one of the challenges because for transporters, inhibitors are more, I would not say easily, but it's I would say, more known, how it's easier to find an inhibitor, let's say, that would increase the interaction, but the modulators that activate transport, it's very challenging because it means that we have to make the transport happening faster. And so I think I would go maybe to one of those lipid protein interaction sites, because I think it would be one of the ways to try to activate transport, but so far I haven't tried anything yet, because I think that we first need to understand the transport process on entirely before trying to modulate it in any way. But that's one of the goals, yes.
We did not.
We have time for maybe one more question, and then we can go to a brief break.
We have a question about sodium binding. Isn't a sodium ion co-transported with creatine? And we want to, I guess the question is how much the sodium binding would affect these models, right? Because this is a symporter, so it brings in the sodium and creatine at the same time. And I'm not sure if these are bound in the, I assume that these are bound to sodium in the structural models, but I haven't looked closely. Do y'all have any ideas about the role of sodium in conformational transitions?
So for the creatine transporter itself, I haven't investigated it yet, but I've read a lot of literature on the serotonin transporter, since there is a lot of information on this transporter. We have crystal structures and so on. And it seems that the sodium ions are there to stabilize the outward open conformation before the ligand comes, and then to facilitate the conformational change for the transport of the substrates. And I assume that it's the same, similar for the creatine transporters, as they have the same fold and same transport process, it seems. Yes.
In our case, when we did the steered molecular dynamic simulation, the sodium ions were present there. However, I don't know if they move with the creatine downstream or what is actually their situation. Mm-hmm. But of course, I'll ask this question to my student, and we will know the answer very soon.
Great.
Thank you very much.
Thank you.
Thank you.
Thank you so much, everyone. I do want to, special shout out to these people, just because this is the first ACD symposium that they're a part of. So warm welcome to all of you, even though it's after the fact. We're so happy to have you here. This is exactly the